it's 3 o'clock and we will start one of the last sessions, Radioligands. And I will start with the introducing of myself. I'm medical oncologist, Elīna Sīviņa, and I'm working in Riga Stradins University and also in a Latvian university and also in a Stradins Clinical University Hospital. And today I am with...
Hello, my name is Donatas Vajauskas. I'm a radiologist working in Lithuanian Health Science University Hospital Kauno Klinikos. I'm glad to share today's event with you and with my colleague. And we'll invite the first speaker. Okay. As in modern days, it's usually, to use ChatGPT. So I try to use the ChatGPT with the introduction of our first speaker. So it's my great pleasure to introduce you to Dr. Thierry Stora, physicist with the Internal Atomic Agency. Stora brings with him wealth of expertise in nuclear physics, particularly in the production and application of radioisotopes, which are critical in both medical and industrial fields. His contributions to advancing nuclear technology for peaceful use have had profound impact on the global scientific community. Today we are fortunate to hear from him as he shares his insights, which promise to be both enlightening and inspiring. So please join me welcoming Thierry. Is it true what ChatGPT writes about you?
Probably 50%. Okay, so welcome and thank you for the opportunity to give you an introduction of this very important session on radioligand therapy. So I will, of course, I'm a physicist, so I will not tell you anything about medical treatments and things of this kind. But I will show you actually how physics and their very fundamental physics could have an impact into development of the field as a whole. And to remind you, actually we will be probably discussing and exchanging on different ideas today, and this was all originating from a bright physicist and or chemist, because Marie Sklodowska-Curie, because we have the Nobel Prize weeks this week, had actually 2 Nobel Prizes, one in physics and one in chemistry. And that of chemistry was actually for the discovery of radium. And you are probably well aware that radium has quite an important role in the field of radioligand therapy. And very early on, actually, when radium was discovered and basic chemical properties were discovered, her husband Pierre Curie, together with the German medical scientist Friedrich Giesel, actually tried to see how this could be useful, and they put a little bit of radium on the skin, and you can see on the upper right the origin of precision medicine, which is you can see what you're doing and you can localize the treatment so that eventually you cure. Of course, it's probably a bit short story because the use of radionuclides for proper treatments is a bit more complicated. And this, anyhow, was the origin and was the development of the field of nuclear medicine, which slowly, or not slowly, but with the phases went from diagnostics to a treatment modality, and the precision of this domain is actually associated with how you can precisely deliver these radio elements to the localization where you want to have the effect. And always when you will have nuclear medicine treatments, you will always associate that to some imaging and checking the efficacy of your treatment. And one of the examples was the first marketing of radium, which was discovered more than a century before, which was actually proposed and taken up by Bayer for more pain palliation treatments than real remission or impressive survival increasing, which then is marketed as Xofigo. So actually, this is already we speak of radium, and actually there is in the scene a lot of other elements, chemical elements, radioisotopes, which will have an effect and can localize or have a precise effect, depending, of course, on how you can investigate the biological target and the eventual internalization of your radionuclides with the tissues you want to treat. And here that's an example which is less advanced, but which is still coming from radium, but you see that's the beauty of radioisotopes. If you have one element, it will become a different element by the decay. So actually the radium you will not inject directly to 2,4, before it was 2,2,3, but this you can be used as a generator, as a kind of a cow that you milk away so that you generate the lead-212, and this itself, you see on the left-hand side, in the decay of the product will be emitting an alpha particle. And this has a great advantage because the alpha particle has a very localized or very precise, because we are in precision medicine workshop, localization of the radiation. And this can have much benefit, even respect to benchmark treatment that you would have with radionuclides, which would have maybe a less localized deposition. So the full purpose of the field of this radiodiagnostics or radioligand therapy is that with the same ligand, you can associate two different radionuclides which will have two properties. So either a diagnostics property, and the most common one would be scintigraphy or SPECT, but you can associate it with a positron emitter, and then in that case it will produce a PET CT scan, which of course is very much on the rise and can be quite quantitative and precise. Or you associate it with a treatment radio elements, which then will have an effect on the evolution of your disease, and eventually would lead to the local necrosis and remission over some of the disease you want to treat. So this field, of course, is we only have one session in this full Precision Medicine Network meeting, but is expected then to receive an increased market share of more than 10% growth overall over the coming 10 years. And we start to see, we have colleagues, of course, from Novartis, which is very much active in this, and we see that some of the drugs might become actually blockbusters in this field. And I'm a physicist, so I have nothing to do with all these treatments, legal approval of drugs, etc. But we are promoting at CERN the use of new radionuclides in the field, and for that we need new techniques, very sophisticated machine, and we need to be able to translate those with multidisciplinary teams, so that it goes from purely test on animals and cells up to early clinical phase. And when you have large clinical phases, as you know, this is very much involving multicentric and medical doctor settings. And I've mentioned already a few of these radionuclides, but you see on this slide that actually you may benefit from radionuclides which have a lot of different chemical properties, a lot of different half-lives, and a lot of different decay properties. And all this combination can lead to very different type of behaviors in terms of either imaging, or if you're using, for instance, targeted therapies, you know that if you use antibody fragments, the real accumulation in your tissues may take from 1 to 2 days, so you cannot work with something which is, for instance, fluorine-18, which is 2 hours half-life to do your PET CT. That doesn't work. Even gallium-68 is too short-lived. So you will need to find another PET tracer, and there is actually a few which are available, but you need to know that they are available and you need to use them. So all that I will focus, of course, more on how we can produce this radionuclides, because on the contrary of a chemical compound or even of a peptide or protein, we need quite big machines to produce those radionuclides. And in itself, this is a field, and there is in Europe actually very competitive facilities which have the capacity to produce these diverse radionuclides. So I will, of course, give you an example of what we do at CERN. And CERN, you probably know more the facility because we have been discovering particles and we are constructing very big machines. But at CERN, we are as well producing radionuclides for a very long time. And this is when you have this diagram of all the accelerator complex that you see on the upper left side. Actually, you have in green Isolde, Rex, I, and Medicis. And this is where you have actually already radionuclides which are produced, and they are produced before Medicis only for nuclear physics, Marie Curie, Pierre Curie, etc. But now very much for serving biomedical research. It's a new facility. It's less new because it's already 5 years old. But this is giving you on the right-hand side the ideas of the different radionuclides we can produce. And I will not tell you what are the beautiful properties of these radionuclides, but I can tell you already on the number 6 or 7 which are shown on this slide, they have all different type of properties, either for chelation to synthesize the bioconjugate, to have an effect on what we call an alpha emitter, on an Auger emitter, to combine a PET imaging with a treatment isotope, etc. So all that is nice, but you need to dispatch, right? So you need to deliver this radionuclides. So the production is not enough. I will show you that we have actually interesting technique to produce these radionuclides, but you'll have at the end to deliver that. And I will come to that in my last part of the presentation, is how you can make a full consortium in Europe which is able to promote these new radionuclides for use for biomedical research. Early biomedical research, again, preclinical, proof of concept, and hopefully some of phase 0, phase 1, first in human applications. And here, just to give you an example, so I'm a physicist, I'm very much about what we call isotope mass separation. So instead of doing radiochemistry, what we do is we separate selectively isotopes by their mass number. And if you're all familiar with nuclear medicine and radiopharmaceutical, you know we speak about technetium-99, fluorine-18, etc. So this little number, 18, 99, etc., is their mass. And actually it's very good because at CERN we know that if you have a mass, you can separate an isotope according to the physical mass. And here, this is a diagram showing you that we can co-extract radium-225, radium-224, and radium-223. This is these little valleys and peaks that you see on the bottom part. And you know, radium-225 is actually a mother isotope to create actinium-225, which is the most demanded isotope for targeted alpha therapy coming up on the market. We have radium-224, which is the mother isotope for lead-212, which is the second most demanded alpha emitters coming up on the market for this targeted alpha therapy. And then on the final is radium-223, which is actually already marketed as Xofigo, as you know, with all the good properties. So why CERN would be involved in making radioisotopes? So actually there is a long tradition not only to do pure and fundamental research, but we have as well exploiting our technologies so that you can go for a medical application. And here I give two examples. So we have, for instance, element selective type of X-ray machines called Medipix collaboration. You can Google it and you will see it. But as well, you may not be aware, but Dave Thompson was the co-inventor of the PET-CT, and was a physicist at CERN, and he was doing that in the Geneva Hospital, which is 5 kilometers across the city and maybe 2 hours because of traffic jams. But to say it, so we have this close proximity with major university hospital, and this is how you can really get this collaborative project. So here it's an example. I don't see how I can make an animation, so I will not do the animation. But this is to show you, we have a calcium analog, which is a positron emitter. Ah, I see, yeah, so you can do one, thank you. And you see simply that you can follow then this metabolization in, for instance, in bone carcinoma, osteoblastoma, which then can give you information, quantitative information on how this barium analog can be metabolized in bone. So next slide, I will go. We have enough. So as I mentioned, so we are now have launched a PrisMap project, which is then a network of facilities which not only produce the radionuclide, but can redispatch that to a research group which are applying to our services. And this is across all the production, purification, dispatch, and you see already the complexity of this, but this is working. So it's complex, but we have this in place and this is working. And here this is giving you an example. You can again go to prismap.eu and you will see that there are now more than 30 projects which are benefiting from our services and which are developing from proof of concept, new radiobioconjugate to, for instance, comparison of Lutathera, for instance, with the same dotatate, but if you change lutetium with terbium-161. And I don't tell you what is the result, because this is ongoing research, but some of these results are coming up. So I'm soon to the end. I see that I'm getting to the last point of the time, and then the presentation. So again, because this is the first presentation of the session, and you will have much more coming up for the full chain of what is used and what is coming up in treatments. But to see that here very upstreams with this Medicis project at CERN and the PrisMap consortium. So we go from you can call it very innovative, but I would call it from the first real interesting radiopharmaceutical which came on the market, and which already by itself is a tour de force, because you see all the hurdles of logistics, licensing, and approval with a lot of variety. I'm sure we will hear that later in the session, to license these products in the different settings in Europe. But we are definitely, with this program that we are just expanding the portfolio of these radionuclides so that you have a more precise match of the physical properties of the radionuclides with the radioligand you want to develop. And one example is, you know, this fibroblast activation protein target, which is very much on the rise. And the difficulties, you have a very short residence time in your tumors of the radioligand. And here we are just proposing to match better radioisotope, which has a shorter residence lifetime, if you want, so that you can have ambulatory treatment, because the isotope will just decay much shorter time than if you would be using a too long-lived radionuclide. So that's one of the examples, and this is ongoing and this is continuing. So we have more than 30 projects coming up. This you can get information either on prismap.eu, and if you're interested, you can really join the user forum. We'll be going to ENM, to the European Association of Nuclear Medicine Congress in 2 weeks' time in Germany. You can as well look at medicis.cern of what we have been doing with this new radionuclides with a small collaboration. And with that, I thank you for your attention.
Thank you, Professor. And we have time for questions. Is there questions from audience?
I have a question, if I may. From your standpoint as a physicist, which radionuclides you see more promising in therapeutical aspects? Alpha emitters, Auger electrons, beta emitters, and why?
So I think very likely you could expect to have a few alpha emitters, to have a few Auger emitters, and to have a few combination of imaging and these treatment isotopes. So there will be not one, not two, but you will have probably a portfolio of 20, 25, which will be able to advance the field in the years to come. So, and this is very much different to what you have today, where this isotope is incorporated in the supply chain from the big pharmaceutical companies, which are very active and very good in the field, but which then is a very different strategy than what is proposed for European researchers, which is the fact that they can access a much bigger spectrum of radionuclides. And this we hope this will be visible in the 5 years to come, because we have launched this PRISMA program. We have seen that there are results which are coming up, and normally that should translate into, I wouldn't say the market, but let's say really the clinical application. Hopefully, as you see, the timelines is 10 to 15 years, right? From really early research to really a clinical application. So you can expect that in 10 or 15 years, there will be a change of the rationale of how the radionuclides are used in this field.
And what about supply chain? We have a known bottleneck, how to go into the therapy on medical setting, what do we need for the facilities, specialists, and etc. So what about supply of such an isotope?
So I suggest you organize a session of supply chain next year, because the answer will be so long that so there are many issues in the supply chain, and Europe is not the simplest playground, I would say, on this. But we are trying to improve things, yes.
Thank you.
Thank you. So let's move on, and I would like to introduce the next speaker, Maija Radzina. She is associated professor. Also, she's working in both universities, and also we are working together in a post-Stradins Clinical University Hospital. So the floor is yours.
Thank you, dear chairs, for a kind introduction, and this is a very challenging talk because innovative, but on the other hand also this is more deeper into the expansion of nuclear medicine beyond the diagnostics into the treatment opportunities. So just a brief reminder of one of the main aspects that I will try to cover is theranostics. This is when we are targeting the possible pathological tissues with a diagnostic tool and also with a therapeutic tool. But ideally, they match, and then we can understand that by the selective glowing of the tumor cells, we also can target them more selectively, which brings it to the topic of precision medicine. And of course, in each case, it could bring it to the personalized management. So theranostics has been quite emerging field in last years, and had an opportunity also to visit one of the international forums this week in International Atomic Agency about dedicated specifically the PET-CT imaging, and all over is a topic about this Lancet publication about the trends and maybe the future perspectives of radiotherapy and theranostics. We know that we can treat tumor differently. With the chemotherapy drugs, we can use a radiotherapy, but we can use also the radioligands, which are labeled with a specific additional tracers to reach the specific cells. And it is unfortunately the main obstacle why it has not developed so further and so expansive throughout the global arena. It is the expense and also therefore availability for these therapies. Of course, as my previous speaker mentioned, it requires a lot of research beforehand. And I must say that from medical point of view, it holds a lot of experimental arena with a variety of the treatments. And therefore, currently, mostly the indications for these theranostic therapies are targeted already for end stage and advanced stage oncology patients. Also, the other part is, as you mentioned, the logistics production happens in one place, but then we need to bring this specific radioisotope into sufficient activity to the patient, rather than bringing patient to the production site. So this was also discussed this week, that unfortunately, if there is no reimbursement, it equals to the no access for the patients. So it's important that we are building the credibility of those drugs to be reimbursed. And these treatments are not as cheap, but they need to be done precisely. You cannot stop them on a halfway. You cannot do just one dose. You need to fulfill the full course of it. And also, it requires a certain setup of the terrain, which means involving and also teaching the workforce, which should be involved in so-called theranostic centers, because it is part of the treatment. And it requires a certain infrastructure. It requires the knowledge of physics, of chemistry, of dosimetry, on waste management, and the safety for the patient as well. And of course, minimum requirements are in the years, and it is already labeled that it requires minimum for physician of the nuclear medicine to have in their competence 100 procedures per year. So within smaller communities, it is quite challenging. Therefore, it's important that we look for collaborations. So clinical indications currently and evidence are in a variety of organs. As we all are aware of radioactive iodine therapy for thyroid diseases, also bone metastatic treatment with a variety also of radionuclides, as you can see, not only radium, but also samarium, and also the specific groups of neuroendocrine tumors and also prostate cancer, and going into the liver cancer as shown also before. So now are the most latest targeted groups. I would say that the neuroendocrine tumors are a relatively smaller group, but of course we want to target the larger populations, the long-term diseases in understanding of the treatment, where we have an opportunity to take the phase 1 treatment, phase 2, phase 3, and we understand that we have already tried everything and there is nothing left to do. And then comes in this theranostic therapy, including the lutetium, PSMA, addressed treatment for castrate-resistant prostate cancer. And as you can see, it improves the survival parameters, even in those cases where everyone would say it's really stage 4, it's still progressing, there is really nothing to do, but these targeted therapies can bring us forward quite a lot. These are diagrams from this Lancet publication, and this is the availability of treatment, annual use of this radioactive iodine for thyroid cancer. I will just go through some diagrams. This is for bone. As you can see, much lesser extent. So thyroid is really nicely covered. The gray areas are of non-available data at that time point of the publication for LIAA. And here we come for neuroendocrine tumor. It does not look much better than for bone, but the neuroendocrine tumors are really complicated for treatment, and this is a long-term oncological disease with a very variety of the expression of somatostatin receptors in their origin, and also in a very variable treatment options for the patient, starting with, of course, mechanical surgery, but also ending with this targeted therapy provided by radioligands. Clinical data for use of gallium dotatate has shown that it is clear an indication for the patients where they have this somatostatin receptor overexpression, but also not only with confirmed cases, but also suspected cases. Sometimes our imaging cannot reveal the full expansion, or maybe sometimes we have full understanding that there should be a disease, but we cannot locate where exactly this tiny, small lesion is. So we need a targeted imaging, and for this we use a dotatate gallium diagnostics. Later on, with the proof of international trials, phase 3 study called NETTER-1 brought an indication of possibility not only to diagnose but also treat the patients. So here we have also the indication for PMNET in adults, and also, as you can see, it's not always the same radio ligand that we use. We can use, in this case, 177 lutetium, but also binded with the dotatate. So we use a different emitter, we use a different radioactivity patterns, but we label it with the same ligand to reach the specific target groups. And as it was proved that there was an improvement in overall survival data, and with a delta of 11.7 months, and also there was an additional studies to prove the concept, and it was again repeated in an analysis of the safety. And we always must keep in mind that there are not only effects, but there are also adverse effects, and unfortunately patients may also develop the side effects, and the side effects usually, of course, is nausea, fatigue, but for all of the toxic agents would apply. But unfortunately there are also negative effects on the salivary glands, with the predominance of dry mouth feeling, and also of course anemia, lymphopenia, which brings into the deterioration of the quality of the patients during the treatment. Lutetium itself may benefit also for other indications, and the somatostatin receptor expressing can go beyond the typical locations of neuroendocrine tumors, reaching out to the small cell lung carcinomas and also glioblastomas. Prostate, with lutetium targeted PSMA therapies. We know that the prostate cancer can express the PSMA, and this binding efficacy can bring the radioligand diagnostics and therapy. So this is the lutetium PSMA and another agent, actinium-225, use in a prostate cancer. As you can see, that again, the presence of the disease is very wide, but the possibility and access to such a treatment is relatively limited. So there were quite nice studies starting again with gallium as a very good diagnostic agent, again binded with this PSMA, this time 11, prostate cancer specific agent and antigen, and prostate-specific membrane antigen positive lesions were, of course, glowing up and showing good results. But then it's not only about finding the disease, but it's about the treatment. When there are multiple bone metastases, when there are multiple lymph node metastases that we cannot irradiate such a large volumes of the body tissue, we cannot anymore treat the patients effective with targeted therapies. This is one of the solutions, and it is already FDA approved for therapeutic purposes, as you know, but it requires certain treatment cycles. Overall, survival rates and of course the risks and are assessed in multi-center trials, and these therapies significantly prolonged those overall survival and recurrence-free periods in subsequent months, as you can see in the diagrams. So there were also safety, again, always is addressed, and there was an addition said that increased drug-related adverse events, there was no data given. To establish the costs of each treatment, it is important to define how many cycles per clinical indication would be needed. So it's not just one-time drug, you need to repeat it, and every time you need to scan again what was the effect of the previous treatment sequence. So sometimes it varies from 4 cycles, maybe up to 6 cycles. So in general terms, in this document, the LIAA counted only 5 in general. So this is one of the clinical cases from Stanford where they used this Pluvicto, this is lutetium PSMA treatment, as you can see, the spreaded disease all over in the different areas, and after cycle 1, the lesions are still there, but they are not as active as they were in the first assessment, and then with the subsequent cycles of treatment, we see that the reduction of activity in those specific targeted tissues. Of course, all of these PSMA-expressed tissue are not only located in prostate-specific tissue or metastasis, but also in the other organs as well. As you can see, there are also ideas of monotherapy or combined therapy. In all of the possible oncology diseases, we try to combine our forces, not only to find only one miracle drug, one miracle solution, but to combine. And in the majority of cases, combinations are always giving longer survival rates for the patients. Also, we can see that these combinations may also be among the radioligand therapies, actinium and plus lutetium, both with PSMA antigens. This is one of the clinical examples of fluorine existing, as we use the fluorine 18, but labeled with DASA antigen for the, in this case, glioblastoma treatment. Here we see the expression. But this time, it is not only used for treatment, it is used to assess the efficacy of treatment. Glioblastoma is one of the very complicated diseases to assess the efficacy of provided treatments. So this can be a molecular imaging opportunity. As Thierry said, fibroblast activation protein inhibitors are, they call it new kid on the block. They think expression in so many new entities. We are not only addressing prostate or breast or liver or neuroendocrine or thyroid cancers. We can address pancreatic, ovarian cancer, even the colorectal cancer, sarcomas, which are not as good in diagnostic field yet. These also provide the opportunities for novel ideas of lutetium and FAPI labeled theranostic treatments. Here is just examples from the literature for ovarian cancer, thyroid cancer, pancreatic cancer, breast cancer, also the cholangiocarcinoma in the liver, specific entity, hard to treat. And for sarcoma, as you can see, the FAPI agent is adjacent to the lutetium or yttrium or samarium, so you can use the same ligand but attach it to the different. So very briefly, in the last seconds, about the nuclear medicine trials. If you count all of the nuclear medicine trials, then majority are related to the diagnostics. If you look at the PET-focused trials, they are all about the therapy. This is a list of existing trials in 2024 in the different phases. As you can see, majority are in a preclinical, different labeling, different targeted cancer groups. So range of the isotopes that are existing can expand into the things that Thierry Stora said, with a wide range of portfolio, even new radio ligands like scandium, terbium, astatine, and so on. So we did also questionnaire to identify the key players within the PRISMAP consortium, and this is what was the response. Yes, now we work with lutetium, gallium, we will move into actinium, copper, and gallium will move to only third place, most likely. So development of those services needs a collaboration in between the production sites, researchers, those who research, also preclinical and also clinical entities. And then we can reach the phase 1, phase 2, phase 3, and this is the arena of existing partners in this field of research. Predicted global nuclear medicine market in theranostics will be only growing, specifically in radiotherapeutics. As you can see, the gray bars are higher. And the future steps are in a necessity to reach the good research communities, to digitalize the data, and to write the good guidelines. Unfortunately, only few percentage of a global end-stage phase 3 clinical trials are done in a smaller countries, in the lower income countries. Unfortunately, they are driven only in the larger ones. So it's important to set up the right centers, theranostic centers. There are written guidelines how to do it the best as a state of the art. And the next speaker will lead you more into the understanding of how to build the multi-stakeholder roundtables to bring this idea of the radioligand therapy plan on a national level or maybe European level, and there will be examples from our Belgium teams of this teamwork. So this is a multidisciplinary topic: radiochemistry, physics, oncology, radiology, technologists on board, radiographers on board, and specifically, of course, the nuclear medicine specialists, physicians. So theranostics will be beneficial for patients, for physicians, and of course for those who are funders of it. With this, I want to conclude my talk. Thank you so much.
Thank you, Maija. A short question from audience. I would like to ask one question. You said about these theranostic clinics. What is your opinion? Should we collaborate in the Baltic states between countries and build 1 or 2 clinics, or should we make our own theranostic center clinic in each country?
This is an excellent question. I think partly, I think the majority of the cases, if we are targeting the frequent incidence cancers, probably these could be addressed in each country, like prostate cancer, and so that would not be fair only to organize in one center. But definitely the more rarer solutions, or if we know that the selection of the patients would benefit to be combined in a time-wise, and we know the expenses of the specific radio tracers and maybe the complexity of logistics would be beneficial maybe to build the excellence centers into the specific areas, like neuroendocrine tumor, or maybe these therapies could be more Baltic concept treatments because of the rarity of the incidence and maybe specificity of the disease.
Thank you.
Mm-hmm. Thank you so much.
Thank you.
So I will ask the next speaker, Professor Donatas Vajauskas. He's head of the Nuclear Medicine Research Center at Kaunas. Please, the floor is yours.
Ladies and gentlemen, good afternoon. Big pleasure speaking here. And I will address more on application of theranostics itself, and I'm going to talk about the targeted radioligand therapy, about lutetium PSMA therapy for prostate cancer patients. So if we take a look at the treatment options for the patients with the prostate cancer, the majority of current options are non-targeted. The targeted therapies are labeled in blue, you can see those. And of course, lutetium, PSMA is also called targeted therapy in metastatic castrate-resistant prostate cancer when we have patient post-RPs and taxanes. So if we take a look at the treatment landscape, so it actually evolving towards precision medicine, And of course, the lutetium PSMA therapy is here. However, despite we have this shift, the non-targeted systemic treatments is still form the current standard of care for most men in metastatic castrate-resistant prostate cancer. So why lutetium PSMA is so important here? Why it's important as a targeted therapy? Because we have several unmet needs in many patients, because we have many patients with metastatic castrate-resistant prostate cancer. And of course, when we talk about prior therapies and the usage, we know about resistance, which is limiting us applying treatment options. And actually, lutetium PSMA has novel mechanisms of action, and we cannot talk here about the resistance from previous therapies. The majority of treatment options are non-targeted, associated with a systemic effect here. And when we talk about lutetium PSMA, we have targeted therapy and therefore minimizing the systemic exposure. And currently approved metastasis directly therapies for the bone metastasis, they are limited to the single organ for the bone. I'm talking about radium-223. And again, lutetium PSMA doesn't choose which organ to target. It actually targets the disease itself. And lack of targeted therapies for majority of men with advanced prostate cancer could be solved by applying these targeted therapies, where more than 80% of the men with prostate cancer has high expression of the PSMA on the cancer cell membrane. So, short introduction into therapy itself. So we have two parts here. One part is radioactivity, lutetium. Thierry already told you about these agents. We have beta emitters with a killing effect, and we have small molecule PSMA, which actually targets the receptors at cancer cells. After this targeting, this complex is internalized into the cell and stuck inside. So what does it do? Actually, beta emission from the lutetium gives us damage of DNA strand, expecting to have double break, which is non-repairable, and therefore we can achieve the cell death. And of course, when we talk about the lutetium and the range of the beta emission within the organ, within the cancerous formation, we have 0.7 millimeters, maximum of 2 millimeters path length. So it seems that the systemic effect here would be limited for our patients. My history with the theranostics began back in 2018 when I saw these images. These is images from the SNMI, image of the year, and in red dots you can see the labeled disease. So all these patients had quite a lot of disease, advanced disease, and on the bottom of each picture you can see the numbers. So this is the numbers representing the PSA levels, and you can see the effect of the PSMA therapy. So it was brilliant for me, and I said that this is going to be the future. And as I see now, which way we are developing, which way nuclear medicine is developed, so theranostics is very, very promising area for the future oncology therapy applications. When we talk about the therapy, please remember in these targeted therapies, we're still talking about the theranostics. It means one portion of it, it's still diagnostics. And PSMA diagnostics here is very important. Today we have the updated version of the PROMISE, Prostate Cancer Molecular Imaging Standardized Evaluation System, which addressing tumor itself, which addressing lymph nodes, and which addressing the metastatic disease, as in this example, as you can see here. We have prostate cancer, which could represent it as metabolic imaging T2 disease. We have many lymph nodes up until bifurcation of the aorta, so it means at least N2 disease here, and we have many lesions in the bones, which already gives us M1b disease. So for the staging, this is a brilliant substance. However, we need to think about the treatment as well. And if we look at these lesions here, which has high affinity of our radiopharmaceutical, but these smaller lesions doesn't have such a high affinity. And this is becoming tricky part when we talk about selection of the patients for the therapy. So let me continue with the indications. Now we have one single confirmed with the EMA registration pharmaceutical, which is for the adult patients with progressive PSMA positive metastatic castration-resistant prostate cancer, who previously have been treated with the RPs and taxane-based chemotherapy. So again, PSMA positive, it means that we have to perform the imaging before we go for the therapy to properly select those patients. And if we go back to PROMISE V2, there is a way we can evaluate which patients are good candidates for therapy. So we have to score them according to the uptake, and then we have the patients who would benefit. And this is important. Please remember this slide. This is very important when we go for the precision medicine, when we treat those patients who will respond to therapy properly. Left this slide empty intentionally to keep up with your attention. Okay, so vision trial. Maija already addressed this study. So we have advanced disease, progressive patient with metastatic castrate-resistant cancer, and one arm of these patients received lutetium PSMA, another arm best standard of care alone. And what happened actually after the therapy, 38% of the reduction risk of death, 60% reduction risk of disease progression. So this is truly amazing numbers, giving us insight into this kind of therapy. Later on, when we go into analysis of these patients, how does these patients do? We see that imaging plays here a crucial role, selecting the patient. And actually, visual read rules from PROMISE V2 are appropriate to identify those patients candidates for the therapy. And actually, these improvements in radiological progression-free survival, overall survival, with addition of lutetium PSMA to standard of care, were greater among participants with higher whole-body tumor SUV mean, which means that uptake is higher of our pharmaceutical, with expectation that in radioligand therapy we will have the higher uptake of the radioligand compound, therefore giving the higher radiation exposure of those lesions to the radiation. So what about these patients who are not so well suited for these kind of therapies? We have this vision trial eligible patients, and we have these patients which could be called non-eligible, and you can see here the patient with the liver disease, and one of the metastases in the liver actually does not express the PSMA. So what does it mean? It means that after such a therapy, we're not treating that area. So what should we do now with these patients? How should we address? Because other lesions actually in liver, they have pretty good uptake here. So it might work, but not on all disease metastases. So we'll come back to this later on. But please remember, this was a study comparing the lutetium PSMA versus best standard of care. What else could we do with such a patient? In the standard treatment settings, those patients after first line of taxanes, they could go into second line of taxane. After docetaxel, they could go into the cabazitaxel. And the therapy study actually addressed those patients, looking and comparing lutetium PSMA performance comparing to the cabazitaxel. And again, we have a very nice high response rate in lutetium arm. And if we look at the response rate on the PSA reduction, more than 50%, we see that in PSMA, we have more patients who responded to this kind of therapy. And this is very important. It seems that we can a bit push the boundaries for these kind of therapies, because the first such a therapies in nuclear medicine therapy, they're coming late in a disease stage. And usually those patients are not very prospective patients for that kind of therapy. So it seems that we can push a bit earlier these kind of therapies in clinical setting, giving more chances, more benefit to our patients. And one of the studies I also would like to mention is PSMA-4 study, where the boundaries are pushed even more further. So the patient with the metastatic castrate-resistant prostate cancer on previous RP therapy, and one arm was lutetium arm, another arm was patient changing RP setting. So it is allowed to change you during the therapy from one RP into another RP, and it's proven that this kind of switch of the therapy works on patients. However, what was allowed, the crossover was also allowed in case of progression in this arm. And there were several endpoints, and at the time of primary analysis, treatment with the lutetium PSMA was associated with the 59% risk reduction in radiographic progression of death compared with the RP change. Later on, at the third data cutoff, treatment with PSMA was associated with the 51% risk reduction in progression or death compared to RPs. However, at the time of third data cutoff, the curves came in together, and this actually reminds me of NETTER-1 study, when at the beginning, during first couple of years, we have very nice difference between the therapies, but afterwards, continuing collecting data, those curves come in together with no difference. And why is that? Because of the crossover. Please remember, so from 234 patients in that arm, RP arm, 134 patients, so it's like 57% of the patients have moved to the lutetium arm. So those patients who were progressing, so their condition has worsened and they moved. So maybe this is the reason why we have such a result here. And of course, again, looking at the PSMA performance comparing to RP change and the PSA levels, again, response rate is much, much higher in PSMA therapy arm. And treatment with the PSMA resulted in higher overall objective response rate for soft tissue, excluding bone lesion progression. But again, you see this recommendation that maybe alpha emitters could work better on bone lesions. And based on those two phase 3 studies, VISION and PSMA-4, lutetium PSMA is the only PSMA-targeted agent proven to significantly improve radiological progression-free survival, to be well tolerated with a manageable safety profile in both pre- and post-taxane settings in patients with RP-treated PSMA metastatic castrate-resistant prostate cancer, and in patients who had progressed on latest one RP and 1 or 2 taxanes with addition of PSMA, lutetium PSMA therapy to best standard of care, significantly prolonged overall survival. And this kind of therapy was well tolerated with consistent safety profile. And of course, lutetium PSMA delayed the time of health-related quality of life deterioration and serious side effects relative to control arms in patients with PSMA positive metastatic castrate-resistant prostate cancer. So it seems that that we have very, very nice tool, but what is next? Do you remember those patients which were not eligible for this kind of therapy? How could we do if we could mix these therapies? And the recent upfront PSMA study, which was done on the comparing docetaxel combining on sequential mode with the lutetium PSMA versus docetaxel setting on the metastatic hormone-sensitive prostate cancer. And of course, the primary endpoint was rate of undetectable PSA levels and many secondary endpoints. And if we look here, undetectable PSA rates at 1 year. In combination arm, we have 41% of patients. In docetaxel group, we have 16 patients. PSA progression-free survival was longer in the combination arm. Radiographic progression-free survival, again, better in combination arm. And what is most important here, we are giving the patient 2 therapies which actually have side effects, systemic effects. However, the grade 3 and 4 adverse events were not so different here. So this is very important. We're not over-treating the patient, but giving the patient better outcome possibilities. So I'd like to finalize my speak and say that pushing these boundaries actually gives us better understanding how such a therapies perform. And I'm a true believer in combination of therapies if we can still manage the toxicity of these therapies and they are not increased too much. Thank you very much.
Thank you, Professor. I strongly agree that in the future, these combinations with the chemotherapy or more maybe immunotherapy with the radionuclide therapy, it will be our future. At this moment in Lithuania, is this lutetium treatment available, reimbursed? Are you starting?
No, at this moment we do not have reimbursement for lutetium PSMA.
Okay, thank you. Maybe other questions from audience? Thank you.
Thank you.
So we are moving on to the next speaker, and I would like to invite Jean-Michel. He is a head of public affairs, Novartis, and he will talk about experience in Belgium with this radioligand treatment. Please.
Thank you, everybody. So what we saw was very interesting because we saw the preclinical research with Professor Stora. Then we saw all the treatment options that are available, which is fantastic. But now, of course, we need to be aware that RLT isn't like, to cite Professor, like an ibuprofen that you give. It's something that your healthcare system should be ready for. And that's something I want to talk about today. So quickly, we're going to go talk about the preparers, why we need it, what we did, and then as a blueprint, but let's be humble, and let's see what's usable and what not. So you have seen this slide in other form presented by Maija, I think. So I think the potential of RLT is great and is really fantastic. So it holds a great promise. It holds to be the fifth pillar in the treatment of cancer care. You have radiation, immuno-oncology, like surgery, chemotherapy, and then the new pillar will really be RLT. But of course, yeah, how can we make sure that we balance innovation, access, and future preparedness? How can we make sure that we continue to find new treatments, to make sure that once we have treatments, people have access, but access is not enough. Just having a reimbursement is not enough. How do you make sure that your healthcare system is prepared? And that's a bit the question we were thinking about in Belgium. So, yeah, RLT has many, many challenges. And so we saw it in the preclinical phase. We saw it also during the other presentations. It's all about teamwork, more than I'm sure, than for other therapies. So the question is, of course, how do you keep, and these are all little plates that you keep up in the air, right? How you keep in the air your governance within the hospitals, between, for example, an oncologist and nuclear medicine. How do you go about regulation and reimbursement for this? How do you make sure that we identify all the needs? How do we make sure we have a best service provision? How do we have enough information? And how do we make sure that we foster R&D and innovation? So to keep all these little plates up in the air, it would be very, let's say, arrogant to say, yes, we as an industry, we have it all and we're going to show you how you do it. No, I think we as an industry are only a part of this ecosystem, and we should collaborate with the whole ecosystem. Otherwise, we're not going to get there. And so what we did, so how we made sure that Belgium was capable of embracing RLT, is that we put everybody around the table. We didn't do it, there was a third party, otherwise it would be a bit complicated. But we started the process to put everybody around the table. Everybody meaning the payer, the authorities, that's the payer, the industry, the clinicians, the patients, really basic scientific researchers, and also the experts around the table. Because in the end, if you really want to have a healthcare system that is ready to embrace RLT, you should make sure that also the stakeholders all play a role, because they all have a responsibility. And also, we must have a dialogue. Before entering into this discussion, we often saw that these were all little universes not talking to each other. So they were all very fond of RLT, all really believed in the potential of RLT, but they all were all just in their own ecosystem and not talking to each other. So I think what we can do is to bring them all together and to say, look guys, we really want this innovation, we want to continue to make sure we innovate in Belgium, we also want access, we want healthcare system to be prepared, what should we do? And so over the course of the 2023, we organized 3 multi-stakeholder roundtables to come to a Belgium RLT plan. So what we did is we have 3 sessions. You saw the slide already before. We had 37 representatives. So we had the nuclear, for example, regulator on board. We had the payer on board. We had the industry, of course, but not only us. Also smaller companies were there. We had university centers that can also produce themselves. Some of them. We had the peripheral hospitals. A peripheral hospital in Belgium, you might laugh, but it's a bit of the second line because Belgium is smaller than Latvia, I think. So peripheral, we mean, let's say, the second line hospitals. We had really also the patients, very important, also on board. So everybody was there, and we had expert opinions. So before starting to talk, we asked everybody, what is your opinion on it? How do you see the future? How do you see what is your vision for RLT in Belgium? And what is needed, according to you, to bring this innovation to the patients? So this was the process. So it's a very lengthy process where I think we had, the very important time was in February, where we had a round table where we put everybody together. And it was also a very transparent dialogue, where we also really had a bit of clashes between, yes, I agree, I don't agree, yeah, you should take more responsibility, no, you should take more responsibility, but at least we were all in the room, and some might say, like, I heard today, we were locked in the room and we threw the keys away until we came up with a solution. Well, this is what we did, and the solution was this RLT action plan, which was then presented. So, of what is it constructed? So actually the RLT action plan has 6 main provisions, like I said. So governance, how are we going to make sure that we include the guidelines, also between oncologists, like I said, and the nuclear medicine. How do we make sure that everybody has a proper remuneration? How do we make sure that we map the RLT needs? How do we make sure that we map what is required for workforce and infrastructure? Very important, of course, because it requires really specific workforce and specific infrastructure. How do we make sure that we have the data? Because if you really want to have a good reimbursement and value for your money, then you also should start collecting the data. And how can you make sure that you attract clinical trials in Belgium? And I think that's also the ambition of everybody here. So based on these 6 different blocks, we had different solutions which were identified. So, for example, I'm going to take out one of them, meaning if you take service provision. Yeah, so of course, when you look at RLT, you need a short-term planning and you need the management of demand and supply. That's something we need to put in place. So, meaning that everybody should make sure that we have knows what the short-term planning is, and also how we can make sure that there is no, let's say, waste of this precious material. Like we all know, supply is going to be very crucial in the future. How do we make sure that we bring in future technology evolution, technological evolutions? Because it's a field that will continue to evolve. How do we make sure that we have something that's adaptable and fragile? How do we make sure that we have total solutions, meaning, for example, a very mapping of the patient journey, where we as an industry, but also as a payer, as a regulator, can really follow what's going on and really can follow the tracks of the different patients. How do we make sure that we have professional training and have enough patient supply? So, for example, these are all building blocks that were identified and that should be in the RLT action plan. So what we did is really form a big, let's say, framework where all these building blocks are being discussed. And I think, it gives at least a framework to start with, because RLT is so big, it's so something really for the future. And if you don't start now to prepare your healthcare system, you might have an "I told you so" effect 6 or 7 years later down the road, meaning that you come with even more innovations, like we saw from the slide of Maija with all the other companies, and that your healthcare system says, I can't absorb this innovation. You should have told me 6 years ago. Well, we're telling it now, and let's all work together now, so that we're not ready for the innovations of today, but especially the innovations of tomorrow, because they're going to come, and if you saw the presentation of Professor Stora, it will not stop. I mean, the science is really progressing rapidly. In the next 5 years, he said, new entities will come to preclinical research and phase 0. So this framework was based really on this. It wasn't a solution, and it's also embedded in a more general European call. So if you look at the European recommendations, and that's recommendations from the European Commission, so it's not a third party doing it. So you see that they also encourage a coordinated approach. They also see radioligand therapy as something where we need to strive for EU autonomy, because we might also end up not being able to get or to access this innovation in the future. We need harmonized clinical trials requirements, because also there you see in RLT, you really need multi-center approaches there. We need to stress, of course, the regulatory issues, education and training, and we need to encourage integration of radionuclide technologies, and of course, everything that's to cost effectiveness. So in our plan, or the plan of the RLT, Belgian RLT, we also included this European recommendation. So it's not only a one-off, let's say, only for Belgium. So in the end, of course, it's all about the win-win. So you might see some things on the slide, you say, well, why, on previous slides, like, yeah, it's not, perhaps not usual for industry to go about all these things. Well, I think we must admit that for RLT, it's so complex, like I said, to really implement into a healthcare system. We have to be careful, we really have to strive for this win-win. We must make sure that the patients have access to the most optimal cancer treatment. I think this is really also the basis of this forum and why I think it's a very good platform. We must find clear market access conditions. We must have clarity as industry and a fair reward, of course. But we also want the healthcare budget to be under control. And that's also something we addressed in our plan, for example, with having early dialogue and early horizon scanning. We must also make sure that the hospitals, it's feasible for them. They must have correct reimbursements for the RLT patient care pathway. And also, we make sure that we collaborate to the precious supply that we have. We manage it as efficiently as possible, because again, it's not like an ibuprofen that you can find on the shelf, and you perhaps all know it, but it really requires a very tight supply chain, a very tight infrastructure, and really a constant rethinking of how we're using our infrastructure as efficiently as possible. So that's really really important in this story. So you saw the building blocks of the previous slide. This is the whole action plan. So we segmented it in different, let's say, responsibilities. So you see that for us, the authorities play a big role. Of course, let's say the hospitals and research organizations play a big role. And then, of course, we also have a big role. And we said, oh, look, everybody has its own responsibility. Now, let's get to work. Because if you only have an action plan, that's fantastic. We're very proud of the action plan. But then it's just another report that we can put on the shelf and say that we're proud of in 10 years from now. And say, oh, look, I remember we once did this report. But now we need to get to work to implement this in a country. And so now we are taking the next step in Belgium. So we published a report. You can all download it online. But now we're working on the implementation phase based on these three actors. And so we formed actually a coalition. It was already a coalition, but de facto coalition. Now we really gave it a name. So it's RLT for Belgium, and you see here, it's more than 30 stakeholders who are busy with the implementation of this action plan. So of course you see basic research, SCKSN, that they have, for example, a nuclear reactor very close to CERN, but also, for example, the industry, not only us, but also smaller companies. And then you have the big university centers, like you all know, but then also smaller centers like this one. So you see that it's really a consortium of different stakeholders that really want to implement this in Belgium. So what we did is now we are creating 6 action groups, which you can see here. The RLT Hub, the pricing and reimbursement, group on data registries, on regulations, on education awareness, and RLT clinical framework. And every group has an action charter. So you see now that this is how an action charter looks like. You have, for example, for the RLT Hub, you see the action leads and the contributors. So these people are not paid to be in there, so it's all voluntary. Nobody is there for commercial direct commercial gain, and they all want to then think about how can we now, for example, these different solutions that we identified in the RLT action plan, implemented in Belgium. And so now we're going to start working on it so that we can propose. Actually, we're not proposing to the government an implementation. We are co-creating with the administration an implementation plan. And that's really, I think, something very innovative. So to finish, I think, I don't have to say it, you heard it from the clinicians, so I'm just restating what already has been said. RLT has the potential to revolutionize cancer care. There will be no one single solution, so there will be no magic bullet, not just one reimbursement and then you're done and you can give access to the patients. No, it will really be a multi-stakeholder collaboration, otherwise you won't be able to really implement it effectively in a country, and especially be ready for the future. And that's also the message that we gave to the authorities, and that we're now co-constructing with the authorities is, we're really trying to be our healthcare system really needs to be ready, and that's how we are working on with this action plan. Voilà. Thank you. I'm 30 seconds over time, sorry.
Thank you. Thank you, Jean-Michel. Do we have a question from the audience?
So much. I do have a question regarding the target groups. So you are building the plan for any type of the available cancer treatments, or you are going step by step in a timeline addressing the highest incidence and maybe in a parallelity, maybe only in a timeline. So how you address the different cancer groups for these treatments in this plan, I mean? Because it requires the further reimbursement, it requires budget planning, and it requires the certain actions, and yeah, how you plan it to target the patient groups?
Yeah, so we have, so first of all, it's not an action plan for Pluvicto or for lutetium PSMA. It's really an action plan, like I said, to be ready for the future. So the purpose is really to be ready for any kind of new indication, a new molecule to be admitted, also not from Novartis. So that's really, I think, where we try to transcend the short-term commercial to the longer-term healthcare system readiness. How are we going to be ready for the future? Well, that's the whole discussion we are now into. And for example, for one of the aspects that we are discussing, the hospitals are discussing, is that there's, for example, not enough remuneration for the RLT health pathway of treatment. So if, for example, you treat a patient with RLT, it takes half a day or 3-4 hours, and that's one consultation. So that's nothing to do with Novartis, but that's, for example, something that is a burden, because for a hospital it takes a lot of time and you're only paid for one consultation. So also in the action plan we are discussing with the authorities how we can reassess that. And you see also, for example, that hospitals are gaining then or trying to recuperate some of this funding via imaging, but that's also something everybody agrees it's not a perfect way, because perhaps you're creating an overconsumption on the one side and underconsumption on the one side. So to really be ready for, let's say, also smaller targets for any type, because I think the new one will be in breast cancer, 2000, I don't know what, which will be very small, then how to be ready for that is it's to already have these discussions up front, so that we're sure that when it comes, it can be implemented. But we can discuss this offline.
Do we have more questions? If no, I would like to thank all the speakers. We are on time.
Thank you for coming, and please stay, because in 5 minutes the closing remarks will be held in this